Welcome everyone. My name is Taisiya Bezhaeva and I work for Elsevier for Professional Services Group. And the topic of this talk will be focused on animal models to de-risk drug safety in humans with specific focus on mental diseases. As you can see from the graph below, despite discarding a substantial amount of project during preclinical phase of drug development, attrition due to safety remains high in clinical phases of drug development. What is important to realize that attrition rates are organ specific, with central nervous system being #1 failure in clinical development. And this actually might explain why, when we look on the mental illnesses, there is lack of new drug that being developed, and overall there is low efficacy of drugs that are currently available on the market, despite the very high clinical need. For other drugs, it's also very important to predict physiological side effects. And this is nicely illustrated by the recent announcement from FDA that required to add a box warning on package inserts of montelukast, which is a widely used asthma drug. And this decision was based on increase in serious neuropsychiatric adverse events reported in the post-marketing reports, mainly increase in depression, suicidal ideation, and anxiety. Only limited number of drugs are being tested for adverse events on the mental health during preclinical phase of drug development, and this might lead to overall increase in number of drugs that are being associated with depression that are reported at later stages of clinical phase and post-marketing studies. And from this table, you can see the top 10 drugs that are being associated with adverse events of depression, and you can appreciate that they do belong to a very broad spectrum of drug classes. So why it is so difficult to predict clinical safety outcomes? Well, the concordance between preclinical and clinical safety observations usually analyzed on relatively small and not validated data sets and after very short time periods of drug approvals. And another issue is that the wrong animal models with low efficacy quite often are chosen. And overall, there is, of course, challenge to reproduce features of mental conditions and to translate physiological and behavior changes that can be measured in animals into emotional and mental states in human subjects. So that results in prediction of treatments in humans in very low levels. So how we can resolve this contradiction? We propose to use large data sets, work with solid statistics, and have an agreement on terminology. In the current study, we was working with PharmaPendium, which contains information from FDA and EMA approved documents on more than 3,000 drugs and more than 1 million adverse event reports that was collected over the time span of more than 70 years. And another very nice feature about PharmaPendium that is mainly curated. So all the adverse events, they are coded to MedRep preferred terms. For our analysis, we picked up top 5 animal species that were mentioned in approved documents as diagnostic test for human events, and the concordance between preclinical and clinical safety-related observation was calculated using contingency tables and the likelihood ratio. Contingency tables actually allow us to look for sensitivity, specificity, calculate positive and negative predictive values, and the likelihood ratio, they actually represent the change in clinical risk when the certain adverse event was observed in animals. In addition, we aggregated reports into high-level categories according matter terms. So for instance, the single effects of agitation, aggression, or abnormal behavior in our analysis were grouped to system organ class of psychiatric disorders. In the second part of our study, we performed a systematic literature analysis to find out if we can improve model efficacy. And we was working with Embase to look for animal studies with depression as major focus. And after normalization, with Elsevier TextMining, which is semantic search technology, allows us to extract specific information on organisms, strains, biomarkers from full text publication. The main research questions of our study were first to find what is the animal concordance for psychiatric issues with adverse events reported from clinical trials. Then we were interested to find out if the positive concordance linked to certain animal species that are being used. And in the second part, we zoomed more into animal models that are being currently used, and we check what are preferred species, how we can induce and measure depression. And in the end, of course, we were curious if such systematic analysis of big data can improve preclinical models. So let's have a look on our results. As you can see from this pie chart, concordance event for psychiatric disturbances are not evenly distributed among the species. So rats have the highest total number of true positive concordances, with mice following behind. Rabbits showed very low level of true positive concordances, and we were not able to analyze primates due to low data that is available. At the preferred level terms, when we look on rats as the species, abnormal behavior, agitation, and anxiety have the highest concordance between animals and human for many reported drugs. And here you should keep in mind that the likelihood ratio does not always indicate high predictivity, despite the relationship are actually highly significant. When we look to the literature, we found out that over 90% of all studies involved depression actually being done with human subjects, whereas only 6 looking at animal models. But you should keep in mind that usually studies involving human subjects, they work just with patient material, so we are not able to look for mechanisms of depression. And animal models at the moment remain a crucial avenue if the researcher is interested in mechanisms, molecular and cellular pathways of depression. What is a good model to better predict adverse events in humans? Well, it should mimic the human condition. It should be relevant to the human mechanisms of depression. And of course, it should be able to demonstrate drug efficacy that is similar to humans. Results of our analysis showed that rodents are the most popular models that are currently used to study depression. And when we zoom into top 10 mouse and rat strains, the Sprague Daily rat and Worcester rats are the most popular ones. And Sprague Daily usually selected for different behavioral tests, and it is less suitable for patterns of behavior, as it shows great resilience when compared to other strains. When we look into approval documents submitted to regulatory agencies, again, rat and mouse are the most common animals that are being used for both evaluation of depression as an adverse reaction caused by certain drug and when drug is tested to treat depression. We were able to identify the most popular genetic models of depression, and here the main recommendation to use conditional inactivation as it allows the investigation of spatial and developmental effects. When we look for gender of experimental animals, here even the prevalence of major depression actually is higher in women. When we look on a number of published studies investigating the condition in women and men for depression, it's equally distributed. And moreover, when we look on animal studies, actually over 70% of all the studies, they use male subjects. And here our suggestion would be of why the use of female animals, as it may increase the predictive value of animal models of depression overall. So how we can induce and measure depression? Here we found out that there is a very big technical challenge in separating induction and measuring of depression-like behavior. So usually if drug candidate is tested on a healthy animal, there is a risk to underestimate drug efficacy when it's tested in normal animals, as it is compared to basically placebo effect in a healthy subject. So we highly recommend to first induce depression-like state. And there are several models to do so, and immobilization is one of the most frequently employed stress models for rodents. Yet there is a limitation because actually we cannot regulate intensity of the stressor. What is also recommended to choose chronic versus acute stress, as it shows higher phase validity, and it's basically similar to a buildup of stress conditions that humans are experiencing. How we can measure depression? Well, behavioral tests were features in almost all approved documents for all drugs that provided preclinical efficacy study, and most prominently used tests, they usually consider behavioral despair, as can be measured by forced swim test, anxiety, it's an open field test, and anhedonia as a prominent components of depression. So lastly, another very important question was to look for biomarkers, how we can measure depression, and we stratify biomarkers that can be found in biofluids between proteins and small molecules, and they actually central to several hypotheses of biological mechanisms of depression that are known. And we also look for biomarkers that can be measured within the brain. We stratify for brain regions, and again we look separate for proteins and small molecules. So to conclude, what are the best practices that we recommend that will allow to increase translational value of preclinical models? It's recommended to induce depression-like state rather than looking at healthy subjects. Looking at chronic induction of stress rather than acute, and combine multiple stressors. Also, when you evaluate depression, it's better to look on different aspects and expand the use of female animals, and of course, adapting existing techniques to study females. To conclude my presentation, animal models, they do remain a very valuable source when we're interested in elucidating the mechanisms of disease, finding treatments, but also predicting potential adverse events. There are still a lot of challenging in modeling, especially mental diseases in animals, yet we were able to identify various techniques that are quite efficient in measuring depression in animals. So in the end, such big data approach that we combined with advanced text mining technologies and statistics proven to be a powerful, feasible, and universal analytic method that allow us to assess efficacy of animal models for drug safety. I thank you for your attention, and if you have any questions, please contact me. Thank you.